Dear all students, today we'll talk about passive agglutination. What is passive agglutination? It is an agglutination reaction happens when I am using particles coated with antigen or antibody. These particles could be latex or red blood cells ghost, and it is coated with antigen or antibody according to the test used. So here, as you can see, we have latex bead. These latex bead are coated with antigen attached. Okay. Here in the other photo, we have latex bead are coated with what? Antibody. Okay. We, as I said before, we can use latex or charcoal, sometimes charcoal or red blood cell ghost. This is material. All of this is material can be loaded with antigen or antibody reaction. Examples of passive agglutination tests: we have virus heme agglutination inhibition test, we have rheumatoid factor, we have anti-streptolysin O and C-reactive protein test. Rheumatoid arthritis factor. Rheumatoid arthritis is an autoimmune disease which the patient produce anti-gamma globulin autoantibody which attack human IgG. Okay, so how can we detect these in the blood? First of all, we have to acquire the serum sample. If our suspected patient have rheumatoid arthritis factor, so his serum will contain anti-gamma globulin autoantibody. So our latex coated beads must have the antigen. Which antigen we have? We have here latex coated particles coated by antigen, and our antigen is immunoglobulin G. When the antibody found the latex coated particle coated by IgG, the reaction will happen. We have agglutination, so we have positive result. Anti-streptolysin O. Streptolysin O is oxygen lapile toxin produced by a strain of bacteria called Streptococcus pyogenes. During the infection, Streptococcus pyogenes start to produce this toxin. Our immune system will handle this toxin by producing anti-streptolysin O antibody within 10 to 14 days. How we can detect anti-streptolysin O in the patient's serum? We have patient serum which contain anti-streptolysin O antibodies, so we must have latex particles coated by antigen. In this case, our antigen is streptolysin O. It is a toxin itself. When the patient serum and the latex coated particles found, we will have agglutination. CRP. C-reactive protein is beta-globulin proteins produced in our body, but when mainly appears in the acute phase of rheumatic fever and other diseases, not only rheumatic fever. Mainly it is stimulated and elevated by bacterial infection, inflammation, and tissue destruction. So how we can detect the CRP? CRP is a protein produced in our body, so we will find it in our serum if we have any case from above. So our latex particles must be coated with antibodies. These antibodies must be against the antigen, which is CRP. When the antibody find the antigen, so we will have a reaction, positive agglutination result. Thanks. Have a nice time. Till we meet. Have a nice day. Bye.